Hello all.
Finally, let's touch upon two types of curves fundamental to biosensor characterization. First one is response curve. This is a graph showing the biosensor's output signal, which is shown here as a function of analyte concentration. In this graph, you apply different concentrations of the analyte and plot their output signals. We can see that with increase in the concentration of the analyte, the impedance of the system is increasing. Second graph is calibration curve. A calibration curve is a graph that plots the biosensor's response against known concentration of the analyte. Now, from the response curve for different concentrations of the analyte, from this information, you are going to plot the calibration curve. For example, 100 nanomolar concentration, you achieved this much of impedance. From this graph, for 100 nanomolar concentration, we achieved this much change. So that quantitative value was plotted in this graph. Similarly, the same thing was done for other concentrations of the analyte, and this is how a calibration curve is obtained. The different values like dynamic range, linear range, LOL, LOQ, LOD, every value is calculated from this calibration curve. So this is the most crucial curve or graph for finding out the performance of the bio. So you apply the analyte, you record the response, and you get a response curve. From the response curve, you plot the calibration curve. This was the summary for this slide. So let's meet in the next lecture for further details.